Hi, good morning, everybody. Hope everybody is well today and at home and you can hear me. So welcome to another ASFA forum, another emergency one, let's say, for the coronavirus crisis. So today we're going to try and cover some different issues to the last time where we did a more scientific view on what the virus is, the pathology and so on. So today, what we're going to try and do instead is focus a little bit more on some of the regulatory issues around what happens in a crisis. So today, then, we're going to cover a little bit of background on coronavirus, some new issues that have come up, some special issues. But if you want to know more about the virus, you should check out our other webinar. Then we're going to talk about some of the emergency regulatory procedures that are ongoing and that are available for different sponsors of products, for example. And at the end, we'll just show a few funding opportunities for people who might be developing products for treating coronavirus or different diagnostic tools, for example. So some of the stuff I'm going to show today, because this is normally an internal training, I would probably skip some of the slides, but because this is also going up for external use, I'm going to assume at some stages a limited knowledge of certain aspects. So I will kind of go over a little bit of basics of drug development just to give a context for some people from the outside who may be less scientific or less from a regulatory field, for example. And as in the last time, I've tried to put the references where relevant so that you can trace what I'm talking about. So first up, just a few new scientific things related to the SARS-CoV-2 virus. So this is a topic a bit close to home because for some colleagues and friends who are pregnant at the moment. So just a bit of information about what is known at this stage about COVID-19 and pregnancy. So first of all, there's two general concepts when talking about transmission of a virus. The first is horizontal transmission, which is just your standard human to human or individual to individual transmission. You can also have this concept called vertical transmission, which is essentially transmission from mother to offspring. So there's three main ways that this may occur. Germline transmission, which is where the virus may integrate into germ cells such as sperm and be passed on to the offspring in this way. There's placental transmission, where you can get what's called transcytosis across placental cells, so from the mother to the embryo in utero, essentially. And in some cases, the placenta may also act as a reservoir for some viruses. And you can also get perinatal transmission, which is what happens during the birth process, where you may get vaginal transmission to the baby during delivery, for example. So there is a little bit of data available, but obviously pregnant women is a lesser cohort than the main population, so there's always less patients. There's one paper at least from the Lancet which describes Chinese women, 9 Chinese women who were in the 3rd trimester at the time of infection. They didn't seem to have any increased severity of disease or different course of pregnancy and so on. The babies that were born seemed to be fine. One of them had a slightly low birth weight, I believe, but may have been associated with another pre-existing condition in the mother. There was no detection of the virus in any of the biomatrices that were sampled from these babies, though the data may be a little bit limited as well. I think all these babies were delivered by a cesarean section, so maybe that also reduced the risk of perinatal transmission in this case. However, there is a few other papers that did suggest a risk of vertical transmission, though the sampling in those patients was done quite a bit after the birth, so it may be that they were just infected in a perinatal or peripartum kind of way after birth. So it's not very clear. There was one baby who was reported in the UK to be positive, but her mother was also positive at the time, and I believe it was a vaginal birth. So again, it may be just a perinatal transmission at the time of birth and not reflecting any risk for developmental abnormalities. So I suggest anyone who's interested in that theme, there is a paper by Favre in the Lancet that has some preliminary recommendations, but there is some controversy about that as well. For example, would you recommend cesarean section in an infected woman who may have a higher risk of having complications due to pneumonia issues and so on at birth? So the recommendation would be to follow local advice and to get some information from the literature if you are in that situation. Okay, so just going on a bit of a tangent from there, just wanted to just talk about the difference, for example, between coronavirus and Zika virus. So everyone may remember Zika from about 5 years ago, which is a type of flavivirus, again, an RNA virus that became quite prevalent in the Americas and was a bit of a concern for pandemic, but seems to have come under control. But one of the issues of that virus was that it was causing some congenital birth defects, such as microcephaly and Guillain-Barré syndrome, for example. So the mechanism for that seems to be quite different to coronavirus. In that case, the Zika virus seems to preferentially infect CD14 plus CD16 plus monocytes, so white blood cells basically, and these act as a Trojan horse, which can basically deliver the virus to immune privileged sites by kind of infiltrating tissues or going from mother to fetus in that way. So there is some suggestion that the infected monocytes have different cell adhesion gene regulation, which may facilitate the spread to different tissues, including sites such as neural tissue, which obviously has the blood-brain barrier, which should prevent the infiltration of these tissues. On another even further side note, there was a recent paper as well suggesting that Zika could be used as an oncolytic virus for treatment of glioblastoma, actually taking advantage of this neural penetrating potential. So that's also something to consider. Zika virus, there was also at least one case of sexual transmission of a woman from France who had no contact with any of the regions where the virus was endemic and had no other reason to contact the Zika virus disease except through sexual contact with someone who was infected. This has not been reported for coronavirus so far, but it may be still too early to tell. In this sense, it's worth highlighting that shedding of viruses into fluids such as semen is relatively common. For example, HIV is transmitted that way, and also in gene therapy clinical trials, it's quite common to detect gene therapy vectors in fluids such as semen and saliva, etc. as well. So that's just something to be looking out for. There is another few papers as well coming out talking about potential for neuroinvasion of SARS, however, which may be a little bit more important perhaps. So there was a paper by Lee that suggested that the neuroinvasion of SARS-CoV-2 may actually be partly responsible for the acute respiratory failure and even some of the pneumonia aspects of the virus. So as well as the dry cough and the pneumonia, which are the most recognizable symptoms, there's a lot of reports of headache, nausea, and vomiting, which are also signs of neurological conditions as well. So there's two ways by which the virus may potentially get to the central nervous system. One is called the hematogenous route, which is basically just through the blood, where it may be transmitted and infect, for example, the brain stem or parts of the neural tissue, and then enter into the central nervous system this way. The other way, which is maybe a bit less common, but also still described in the literature, is the neuronal retrograde route. So this basically means, going back to the other side, that the virus can traverse through neuronal connections, for example, to distal sites. So there's two types of transmission: anterograde, which means it goes from the nerve cell itself into the synapse, and the other way would be from the synapse back into the nerve cell, which is retrograde transmission. So this has been reported for quite a few viruses in quite a few situations. For the coronavirus, it is suggested that there may be some retrograde transmission through peripheral nerve terminals such as the vagus nerve, which is the nerve that's responsible for, well, not solely, but partly responsible for breathing, for example, connecting the lungs to the brain. Also other nerves such as trigeminal and also the olfactory bulb, which is obviously in the nose and maybe a closer link to the brain. So some of the data suggests this may be important is that SARS patients did, some of them had detectable virus in the brain. There's also animal models that suggest this neurotransmission, and in some models of SARS and MERS virus, their brainstem was infected. So whether this is actually occurring or not is still to be seen, but it may be an important mechanism for describing some of the serious cases. Also, going back to gene therapy, it's also been proposed that anterograde and retrograde axonal transport of these vectors can be used as a mean to get delivery to the central nervous system as well. Okay, one of the last background papers then quickly was just another report describing the potential that there might actually be two strains of the virus circulating, an L strain and an S strain as it's reported. So there's this paper from Tang that describes the mutation rates and different species based on sequencing. So just to cut a long story short, it seems like there is a very high mutation rate in the spike protein, which is partly responsible for the cell attachment and entry. So there's a lot of synonymous mutations, which are mutations that do not change the amino acid. So they're just changes in the nucleic acid that are just, for example, random or maybe introduced by extreme proliferation and selection, for example. So on the right, you can see here a bit of the sequence. You can see that there is some two SNPs, which are single nucleotide polymorphisms that seem to be linked to these strains. These have divergence from the pangolin sequence, which is proposed to be the sequence of origin, though the bat may still also be the origin of this coronavirus. That hasn't been determined equivocally to date. But what they have seen is that maybe the mutation rate in this protein is very high and may be suggesting that it's not derived from pangolin for this reason. Also, this high mutation rate may also be driven by natural selection. And indeed, looking at the divergence of the strains, it appears that in China particularly, there was a predominance of the L strain at the beginning of the infection, but once the measures imposed by China to stop the spread came into play, there seems to be put a bit of pressure on the virus, and it slowly developed into the S strain, which is more prevalent outside of China and after the lockdown there was imposed. There also appears to be some patients who may have been infected by both types at the same time, though whether that causes more severity or not is not known, but we'll probably see a little bit more information on this and any natural selection that is occurring in the virus. Okay, so that's all we're going to talk about the virus in the background. Now we're going to just go and try and talk a little bit about how we deal with this situation with the virus, and all of these stories here, such as these media reports about vaccines coming out, how long is the vaccine coming out, who's doing testing kits, how all these drugs and trials being regulated, because anyone who knows will realize that these procedures usually take a long time, drug development can be slow. So what does this mean then, that these reports in the media about getting a vaccine available as early as possible? So we're just going to go through that a little bit now and describe the processes that are available and procedures that are being used by EMA and FDA, for example. So just a quick primer for anyone who's not really familiar with drug development processes. So this is just a quick overview of how it works. Obviously, you need to have some kind of target and think, I need to down-regulate this or up-regulate this. You find a compound, you test it in vitro, you work out that it has activity and describe a mechanism of action. You then improve the candidates that you want to go into clinic. The next step is the real important, let's say, development stage. Once you've got your candidate gene, you have to do safety testing in animal models, as well as other kind of mechanistic studies. And then you start your clinical development, go phase 1 with few patients up to phase 3, which is your pivotal trial with a lot of patients. Obviously, at this stage, once you have your equivocal data of safety and efficacy, you present your marketing authorization application or your NDA or BLA to FDA, and they approve your product. And then after that, you keep doing clinical trials. But just focusing on vaccines, just to keep the talk as tight as possible. This table here is showing, so I'll make that bigger. This table here is showing you the development timelines for different vaccines. So on the top, you can see different types of indications. On the left side, you can see the different stages of development: preclinical, phase 1, 2, 3, and the regulatory submission. So here you have the duration in time, the normal time it would take to get a vaccine onto the market. You can see here, most of the time it will be about 8 to 20 years to get on the market. That's from initial concept to marketing. Here you also have the percentage chance of actually having success to transition from the different stages here. So just highlighting HIV, for example, you can see here the percentage chance of transitioning from late stage or even from a regulatory dossier to an approved product is 0 in some cases, because there is no product available. So that's obviously quite alarming in this case, where you think, how do we get a vaccine onto the market as quickly as possible when it takes normally 10 years and the chance of success is very low? So this is exactly what we want to talk about today: how to get around that problem and how to get something to people as quickly as possible to try and reduce the healthcare burden and the toll of death and so on. There we go. Right. So basically then, we're in an emergency situation. This means we need to have accelerated timelines, and we also have unusual regulatory situations in play, which could be many, many things. But you might have, for example, as we've seen, saturated healthcare systems. So how do you do a clinical trial when all of the medical resources are oriented towards treating patients? You also have special clinical trial designs that we will go through very quickly at the end of the talk. And you also may have these situations where it's very hard to demonstrate efficacy or even safety in the timelines that are available. When looking specifically at vaccines, for example, there are certain types of situations that are even more unique that may completely prevent you from developing a vaccine in this case. The first is that maybe your disease or your condition does not even exist, and that would be the case of smallpox. You may still want to develop a vaccine to smallpox, but it's literally impossible to see if it works because there is no smallpox. The other situation, which is where we find ourselves now, is that you have a disease or a condition that doesn't normally occur. So you may have a vaccine, but you can't test it, but then suddenly there's an emergency situation. So you may have to change your vaccine or test it in unusual situations, and then hopefully that disease will disappear again, so then you still can't keep developing your vaccine. The other situation is more common to things like influenza, where you have every year different strains emerging. So you may have a vaccine one year that's useless the next year, so you need to keep changing your vaccine every year. Whether this will happen with coronavirus or not is to be seen, but it may be the case, but we will just have to see. So we're going to focus mostly on vaccines and not small products, but we'll just go through a little bit of the procedures about how you can get a vaccine onto the market in a situation like this, which is an emergency situation. But first, a quick overview of some of the measures that are available at the EMA at the moment for accelerating product development. So the first and most critical one, if anyone is interested, is there is a new fast-track scientific advice procedure at EMA for coronavirus-related issues. In this case, you have to contact the EMA first and tell them what you're doing and why you think you are relevant for this procedure, and they will screen you and tell you whether they think it's worth it or not based on what other projects they have or the readiness of your product and so on. And then you would do essentially an accelerated scientific advice procedure. So if anyone's not familiar with what scientific advice is, it's basically where you'll talk to the regulators about what you're doing and why you want to do it, and they will give you advice about how best to proceed to ensure a more likely positive and successful outcome of what you are doing based on what they expect and the circumstances. Another scheme the EMA has is the PRIME scheme, which is a priority medicine scheme. This is for establishing early dialogue and having constant EMA or regulator presence in the evaluation, and also will give you an accelerated assessment. This could still be relevant now. You might want to start something now, assuming that you will be on the market in a year or so's time. So it's not completely irrelevant, although it's not an immediate outcome either. And a few other things they have are accelerated assessment, which is reducing the time of review of your marketing authorization dossier. So maybe there you have a lot of data. In this case, they would review it in expedited way just to reduce the time. I will show you a little bit about that later. There is also conditional marketing, which means you might get your marketing approval before you have complete efficacy data. For example, you might only have a biomarker or some preliminary data, which you can then complete at a later time. There's also a couple of different programs such as compassionate use, or the FDA calls it expanded access, which is where you can get access to unauthorized medicines for patients with an unmet medical need in special circumstances as well. So this also is used quite a lot in these kind of situations. And also there is a special review procedure related to conditional marketing authorization, which is called exceptional circumstances authorization, which is used essentially only for pandemic vaccine approval pathways. So that we'll show you that in a minute. So just talking again just about vaccines, because some of those procedures may be relevant to diagnostic tests, even medical devices, but the next couple of procedures I'm going to show are just very specific to vaccines. So these are usually only for emergency situations as well, although the modification procedure for seasonal update of vaccines is not always relevant to pandemics. But as we mentioned, it may be that you can approve your product this year and then next year there's a new coronavirus strain, so you may need to do that procedure as well. But the two main procedures for the emergencies are the Mockup procedure, which allows you to approve a vaccine in advance of a pandemic, and I'll show you how to do that. In this case, this is where the exceptional circumstances approval applies. The other way is the emergency procedure, which requires essentially a new dossier at the time of the pandemic and may be based around a conditional approval. So I'll just go through those quickly. So this is the mock-up, or sometimes it's called the core dossier procedure. So if you're looking at this diagram here, you can see there's a pre-pandemic state where, for example, you may be developing a coronavirus vaccine, but you don't know what the pandemic strain is going to be. So you make a kind of mock-up dossier, which includes all the information that you can generate from non-clinical safety studies, manufacturing, and so on. And the EMA will approve that under exceptional circumstances. And then when a pandemic is called by the WHO, which is the current case, then you have to identify the current strain and then try to substitute your mock-up strain with the current pandemic strain. So this usually means that you have some kind of platform in place, for example, for cloning the vector or for producing vaccine antigens or so on. Then you will start to generate your data, whether it's non-clinical, clinical, and as your data becomes available, you submit it directly to the EMA and they review it on a when it comes basis. So this is called a rolling review. So all the information is essentially being reviewed in real time to speed up the process as quickly as possible. The actual time of this is not clear. It depends on how much data you need to generate and if it's coming from clinic, non-clinic, and so on. And then when you have sufficient data, what you would then do is submit a variation procedure. So you would then modify your existing mock-up dossier to then substitute the real pandemic strain for the mock-up strain that was used in the early dossier. The EMA has already then essentially reviewed most of the information. They would then review it very quickly. They would make an opinion to the European Commission, and then they would approve your dossier, and the vaccine in principle should be available within 10 to 20 days from the EMA or the CHMP opinion. There is still a lot of follow-up activities with these kind of procedures, so that would still apply. Okay. The other case is emergency procedure, which is basically when you have no dossier at the beginning. For example, it might be a completely new type of virus or one that no one suspects could be a pandemic, so no one's got a mock-up procedure in place. For example, the mock-up could be used for pandemic influenza, which was predicted to be more likely to be a pandemic than coronavirus, for example. But in this case, you might start now, there's a pre-pandemic, you've got no dossier, and then suddenly a pandemic starts, and then the same situation applies. You might start to develop a vaccine as quickly as possible. You undergo the rolling review with the EMA, sending data and your dossier in real time as quickly as you can. They would review the dossier a little as they go, but then you would then have to submit a formal marketing authorization application once you have demonstrated all of your data. The EMA or CHMP would then review your dossier as a standard accelerated assessment procedure, which takes about 70 days. They would then give you a conditional approval, and then the CHMP would pass the decision to the European Commission, who would then approve presumably the final dossier, and you would get your vaccine onto the market at that time. So just to go through very quickly, what's the difference between conditional marketing authorization and exceptional circumstances? So here you can see the two legal bases at the top, and also we can find additional information on those. The main difference is really here, which is basically exceptional circumstances is you cannot provide any data on efficacy, for example, in the pre-pandemic state, because there's no pandemic to test your vaccine in. The conditional approval might be that you have some data, but it may be preliminary data or not sufficient data, but it's still suggestive of a positive risk-benefit balance. So in the case of conditional marketing, every year you have to renew that authorization just to show that it's still conditional until the time comes when you can make a full normal marketing authorization dossier. With the exceptional circumstances, you still have the annual reassessment, but it's assumed that there is no normal marketing authorization dossier because there is no pandemic at that time. Okay. So the modification procedure, as mentioned, is not really for pandemic situations. It may be useful in a post-pandemic situation, but basically it's just a procedure by which the seasonal flu vaccine, which is the most common case, is updated every year with the new strain as it is detected. So those who get these kind of mails will see that the EMA and FDA, for example, publish the current strain information every so often, and they expect that to be updated in these kind of dossiers. So that's obviously due to mutation rates in different proteins on the influenza that may change, obviously the recognition, antigen recognition, and so on. So I won't go through that in too much more detail. There's also a couple of other procedures. We did something like this quite recently, actually. It's an Article 58 procedure, which is basically a procedure for making medicinal products available outside the European Union. So obviously most of this law that we've described or procedures is for getting products onto the market into the European Union, but maybe, for example, you have another third country where you want to have a product because there's a specific outbreak there or specific situation. So this is when you would use this kind of procedure. So this can apply also to vaccines, for example, those that might be in the WHO's expanded program on immunization. It may be for other public health priorities, for example, target diseases, malaria, for example, is a classic one, or some other tropical kind of diseases that can emerge at different times. I won't go through the details of that, just to keep moving. For the FDA has what's called the Emergency Use Authorization, which is described in Section 564 of the Food and Drug and Cosmetic Act, and that was also amended by the Pandemic Act. And basically this describes what the FDA calls medical countermeasures, or MCMs, which are unapproved medical products which might be of use, or these are approved medical products but with a new unapproved use that might be useful in a situation. And they describe these as the CBRN threats, the chemical, biological, radiological and nuclear threats. So for this act to come into place, the Health and Human Services Secretary must declare an emergency, which they have done, and therefore this enables FDA Commissioner and some of the other bodies to approve products based on the EUA system. So if you're interested in doing this, this is currently active. There is some specific guidance from FDA on this. Again, this is not meant to be a masterclass on this kind, it's just to give you an idea of how these procedures work. But basically, like this fast track scientific advice with EMA, you should contact the FDA center that would be reviewing the product, for example, the CEDAR or whoever it might be. There is a list in the guidance. So do that before submitting any procedure, and they will tell you what the most suitable vehicle to use is, because they may consider that you need an IND, they may want a master file already or something like that. There is also the EUA submission itself, and the guidance gives you ideas about what sort of content is required there. So these kind of applications also generally should include something for healthcare professionals, because obviously these are the people who are probably going to be using the products at this time. So it's always important to consider in these applications who is the user and what kind of information needs to be available to them to keep it as simple as possible. Okay. So the WHO also has a couple of different programs. There is the Emergency Use Assessment and Listing Program. This is used especially for vaccines, for example, just to expedite their availability. And it's a kind of guarantee program to some degree that basically assures that the quality, safety, and efficacy is okay for emergency use. But obviously that means in these situations, things have to be done quicker, and things need to be available to patients and medical staff quicker. Some tolerance of less certainty or maybe slightly less quality to some degree may be possible, but they need to be basically available as quickly as possible. So this program was started in response to Ebola and is different to the WHO pre-qualification of medicines program, which also makes a list of medicines available and manufacturers can put their products there, for example, for availability for these programs. But that's different, and I'm not going to talk about the pre-qualification program today just for time as well. So for the EUAL program to come in place, you need to have a public health emergency of international concern, which is the case. WHO has formally declared that in this case. You have to have a rational use for your product, or there has to be a shortage or no other product available, which is pretty much the case here because it's a new disease. Obviously, the vaccine should be under the regulatory control of one of the national competent authorities who the WHO has qualified. So that might be IMPS in Spain or MHRA in the UK, for example. So they would mediate a little bit the review and revision of the dossier. The product needs to be manufactured under GMP, good manufacturing practice conditions, of course. I have seen that FDA can waive GMP requirements in the emergency situations, and that may also be the case, but it's assumed that there was a previous inspection or so on for the facilities. And I believe that the inspections at the moment are basically stopped for the moment anyway because travel is not possible. So full GMP in that situation may also be compromised. And obviously one of the key things here is that the WHO or the competent authorities would like you to have commitment to continue with the studies. For example, if you get a conditional approval or you get your vaccine on the market in an emergency situation, that doesn't mean it stops there and you don't have to do anything else. You have to keep developing, you have to do post-marketing follow-up, you have to keep improving the quality and the safety of these products once the emergency is over or even as it continues. Okay, so now we're going to just go quickly into more detail about some of the vaccines that are coming out and how they're being tested and so on. So this is some information from the WHO. This is their target product profile, TPP. This basically is a brief list that shows what the ideal product for the WHO for this situation would be. So you can see this, what they would require here. So I'm not going to go through all of that, but some of it is quite obvious, but just to mention a few things. So obviously a single dose would be ideal because it makes it simple, but with vaccines, as we know, that you might need to have a prime and a boost. So 2 doses is also acceptable. That's not a problem. Ideally, they might want to have your vaccine being developed from a platform-based approach, so you have a proven strategy that works, it can be produced and so on at in a quick way. So that's point F as well. You need to be on the market as quickly as possible. Obviously, you might need large capacity as well. You obviously want something that has a durable response, not something that lasts a few weeks and then comes back again. Vaccine stability is a big issue. The WHO highlights here only genetic stability, so obviously propagating viruses in culture to make big batches and to make attenuated vaccines or so on has the risk that you also are making mutations in the vaccines as you prepare them. So that's obviously not ideal and can be more of an issue for some viruses than others. Stability here could also, in my opinion, refer to the stability of the vaccine product itself, because if you have to disseminate a huge quantity of vaccine through a cold chain, that can also be problematic because there just may not be resources. So something that's stable, ideally at room temperature or even just at 4 degrees is better. And obviously something cheaper is also ideal. Okay. So from the WHO vaccine candidates, there is a list on their website where they have already received 41 proposals for vaccines to be developed. So when they were reviewing these, they looked at some different aspects related to the design, what type of vaccine it was. For example, there's basically all the main types of vaccines there: DNA-based vaccines, attenuated vectors, protein subunit vaccines, and so on. A key consideration was whether the technology was part of an existing platform or not. And we'll get to that when we talk to the 3 main candidates at this stage. The other thing they were looking at was the stage of development, but the reality is that all the vaccines except the Moderna vaccine are only in preclinical phase because none of them were prepared for this kind of situation at the time. So Moderna was in clinical phase as of March 13, but that's because they just started a phase 1 clinical trial. So you can see the details on the website, on the clinicaltrials.gov website, if you want to have a look. This is just going to be a safety and reactogenicity study in healthy participants at first, and also we'll look at immune responses. So this product is an mRNA-based vaccine that's encapsulated in lipid nanoparticles for intramuscular administration. There will be 2 administrations, in this case on day 1 and 29. There is also 2 other companies as well as Moderna doing mRNA-based vaccines. One of them is CureVac, who you may have seen in the news because they received quite a lot of funding from the EC, and also they are alleged target from the Trump administration because there were some rumors that they were trying to buy or take over the company to make a US-based vaccine. There is also BioNTech, which has another mRNA vaccine, and they have been linked with Pfizer as well. So just some issues that are coming up with the vaccine development. From a non-clinical perspective, there is what in regulatory terms is called the relevant species, which is the best animal model for studying all these things. And I've just highlighted a few special issues related to relevant species that have come up. So first, we need to see what is the best model to mimic the human infection. This is clearly not known at this stage because even from a human point of view, there is still a lot of data coming in. Obviously, the mouse may be the best model, but ferret is also being developed in Australia, I believe. Ferret is also used for influenza, so maybe that's one of the logical reasons behind that. So some of the key questions there is what This is the cellular tropism of the virus in these animal models. What cells are infected? For example, we mentioned the case of the monocytes with Zika virus. Is that happening here, for example? Also, it may help to identify the species of origin, so to work out where the zoonotic transmission comes from so that measures can be taken, monitoring of that species can be done, for example. There is also a really important concept that doesn't get much focus, but this is basically the possibility for viral-induced enhanced disease illness. So what this basically means is that you can actually enhance the illness of a certain virus by actually inducing immune responses in some cases and in some circumstances. And also on the other side of the same coin, you may do testing in relevant species, for example, with a vaccine, show that you can induce reactogenic T cells, for example, but that doesn't necessarily mean you have a clinical benefit. So you may have an immune response in animals, but that doesn't mean you have clinical protection. But in terms of the viral-induced enhanced disease, what can happen, for example, is you can have antibodies which, instead of being fully neutralizing, maybe partially neutralizing, and they can actually enhance infectivity. So this is called antibody-dependent enhancement. So they bind to the virus and they actually increase the uptake of the virus in cells, so actually increase the proliferation. Unfortunately, this has been reported for SARS vaccines and in SARS, and there is some suggestion that even in early cases of SARS and related coronavirus infection, that antibodies to the virus may actually help that situation. Whether that actually correlates with severity or not is not known, and there's really not enough information about that known. So I think animal models and animal studies of that would be very useful. Obviously, talking about non-vaccine products, animal models can also be used for antiretroviral studies as well. Okay, in terms of clinical aspects, the WHO has published what's called a master protocol for a clinical trial design for testing vaccines. So you can see on the right here, this is actually the cover of the vaccine master protocol, but there is one for small molecules as well, which is very similar. So this actually tells you more or less the overall synopsis of a clinical trial for doing vaccine studies in the outbreak situation, basically. So the master protocol, if anyone's not familiar with that, is commonly used in oncology. FDA has some quite specific guidance on that, but basically it's a situation where you put in quite a few different products for example, an indication like breast cancer or whatever, and you test them to see which one is the best, and they slowly drop out one by one until you get to the answer of your study. And this will be the case here, where you have vaccines will continue until they're either shown to be a futility index is shown, or whether they're shown to have some kind of effect. And in this case, you can actually do the trial in multiple sites and cover as many sites as possible. So the overall design is meant to have a single placebo or control arm. This hasn't been specified yet what that would be. Obviously, a full placebo is recommended in terms of the objectivity and the robustness of the trial, but may have ethical implications because it's a very serious situation, and injecting placebo at that time may not be the best thing. The randomization is 1 to 1 to 1, meaning that all the groups will have the same number of participants in there. So I will show you an example of where this has been applied previously. This is a small molecule case, or at least it's a biologics case, when there is some antibodies there as well, but you will see remdesivir, which is an antiretroviral, which is being tested as well for coronavirus. So this kind of master protocol approach was used in the Ebola virus disease trials that was conducted in Africa. You can see this publication here from Mulangu from at the end of last year, which is quite a timely publication thinking about it. So in this case, they had several sites in Africa where there was the Ebola disease outbreak, and each product was randomized at each site. So each product was tested at all the sites, 1 to 1 to 1 in the same way. The control in this case was chosen to be ZMapp because that had shown to have some benefit in another trial, which was the Prevail 2 trial that was done in Liberia. And this study was performed under an IND, so the FDA said it was okay. It was also approved clearly by the local ethics committee and was done in coordination with WHO. And it showed obviously that I think it was the MAB 114 and the Regen EB3 products were the most effective in this case. They reduced the incidence of death in these patients. So that there is for vaccines, this can also be a little bit of a complication. For example, if you're in the middle of an outbreak, who are you testing with the vaccine? Obviously, in the case of Ebola, you want to be testing the people who are at risk of contracting Ebola. You cannot test people in New York with less risk, for example. But how do you do that? Because if you're in a city or a village where there is an outbreak, how do you do the vaccination schedules to ensure that you're going to have some kind of benefit while being able to show the efficacy? So in this case, one of the classic designs is to use what's called a ring trial design. So here you can see this figure I've robbed from somebody from the internet, so apologies if you recognize your work there. But basically you might have an infected patient, which is the index patient, and then you would work to identify all the people that person has had in contact. So first of all, starting with the closest contact or those with primary exposure risk, and then you would work out the people that may have had secondary exposure to that person. Then each of those primary and secondary people would then be vaccinated in what's called a vaccination ring. So another way they also vaccinate in these cases is to do an immediate vaccination. So detect the patient, introduce them into the clinical trial, vaccinate directly, or to wait a certain period of time and then vaccinate. One of the reasons for doing the delayed vaccination is that those patients in direct contact may already be infected. If they are, you may vaccinate them and then think your vaccine is not working because they become infected. So this is obviously clearly another issue with vaccination in these kind of emergency situations, that the kinetics and the target population is very difficult to choose, and it can be very difficult from a logistical point of view to do. But basically this is how it is done, and this is probably how it would be done in the case of the coronavirus as well. Okay, so finally, just to close off, we're just going to talk about a clinical trial that is ongoing here in Barcelona. So this is a trial that's being coordinated by Uriel Mitjà from FLS at Can Ruti. So this is quite a novel strategy, and it's got quite a bit of media attention in the last couple of days. It's actually a two-sided clinical trial. On one side, patients who are infected with coronavirus will be given Duronavir, which is an antiretroviral typically used for HIV treatment. And the idea there is to reduce the viral load in these patients and therefore decrease the chance of infection. On the other side, those who are in primary contact with the patient will get hydroxychloroquine, which is an anti-malarial product that is used for some types of malaria, but in this case, it is being used to as a prophylactic to reduce the risk that transmission will occur in caregivers or primary contacts, for example. So this seems to be received a bit of attention not only from WHO but also the Bill and Melinda Gates Foundation, which is good. I think there is another similar trial also ongoing in the US, but maybe with just hydroxychloroquine alone. And obviously in this case, this trial has been approved basically within 1 week or so. And this has been possible through expedited review, not only by the local ethics committee, but by the Spanish National Competent Authority, IMS. They will also will continue the collaboration with other partners to look for vaccines. And Asphalion is proud to be able to support them in this clinical trial. So we are hoping to see a good response from this. It will be quite a short trial of about 28 days. So hopefully within a month, we can start to see some positive data from this. To conduct this trial, they're also looking for donations to fund the project. So I recommend you go make a donation to the website that I've indicated at the bottom of the page. Finally, if anyone is doing any of these kind of projects, here is some funding opportunities that are still open. This is checked yesterday. There is a European Commission call that closes today, which may be too late, but there is some newish opportunities for funding for coronavirus-related research for anybody who is looking to set up this kind of project or looking for collaborations. In particular, the DFG, the German Research Foundation, will announce some new information in the coming days. So have a look out for that, as well as the big NIH calls. So with that, thanks for listening. And I don't know if anyone has any questions.